name is Grace Chao, and I'm a first-year PhD student in the laboratory of Dr. Crystal Sol here at San Diego State University. And in our lab, we primarily study an enzyme called isocitrase dehydrogenase 1, or IDH1. And this enzyme catalyzes the reversible reaction of isocitrate, or ICT, to alpha ketoglutarate, or AKG. And it does this by using NADP plus as a cofactor and converts it to NADPH along with some good old carbon dioxide. Now, this reaction primarily happens within the cytosol and peroxisome of cells. So, peroxisomes are these tiny little intracellular molecules that have a multitude of different functions. So this is a blown up image of a peroxisome. And they include the breakdown of very long chain fatty acids, the degradation of hydrogen peroxide, so it plays a role in ROS and extra oxidative stress, and the synthesis of ether lipids. Now, mutations in IDH1 typically drive a variety of cancers, most notably low-grade gliomas and glioblastomas. But interestingly, while most IDH1 mutations actually ablate this particular reaction, so you have an IDH1 that is mutated, and this reaction does not occur at all. Some IDH1 mutations actually generates a neomorphic, or a new and different reaction, and converts alpha ketoglutarate to an oncometabolite called D2-hydroxyglutarate, or D2HG. And this reaction actually uses up NADPH to generate NADP+, which actually depletes NADPH from cytosols and peroxisomes, which is necessary for a lot of the pathways involved in these functions. So I'm interested in studying what the intracellular consequences are of these IDH1 mutations. And I can do that by looking at cells that express wild-type IDH1 enzyme and cells that expressed mutated IDH1 enzymes. And by doing so, I can assess the different reactive oxygen species to see how stressed they are. I can look at different lipid concentrations to see if these pathways are disturbed, and a multitude of different other metabolites. And by doing so, I can illuminate the different features and consequences that IDH1 mutations have within the cell. So that is what I will be doing as a soulmate. If you're interested in listening or learning more about us, please check us out on Instagram or Twitter at The Soul Lab. Thank you for listening. Did it stop? Oh my God. No, it's not stopping.